Why are soils important? There are all these soil-based ecosystem services that we depend on. This is part of the big natural capital ecosystem service push that's going on at the moment globally, and we get all kinds of things from soils. We get the kind of supporting services we're familiar with: soil formation, primary production, nutrient cycling, provisioning, of course, for food, fibre, biodiversity and platform functions like building on, climate regulation, and cultural recreation, heritage, and cognitive. And I think we can pretty much see that we're getting most of those coming through at Knepp one way or another. Why soil biology? Why does it fascinate me? And I've spent years doing soil biology, and in the context of land use, really a big focus on ecosystem restoration. We know the importance of soil biology and organic matter decomposition and nutrient cycling, diversity, ecosystem variability and productivity, and it's called the biological engine of the earth. And there's lots of evidence coming out at the moment that soil biological communities can control the assemblage of vegetation's diversity. There's some work we've just done in the Netherlands has shown that very, very clearly, that if you establish the right biological community, you can actually start to control what vegetation comes in. How much is there? Well, in a handful of arable soil, which is very low in terms of the soils that we've got here in the natural and semi-natural systems, there's about half a gram of fresh biomass, over 10,000 species of micro-organism in a gram, and that's a conservative estimate. So that's equivalent to 5 tonnes per hectare, about 100 sheep below the surface. Imagine that they were all sort of bubbled up to the top, the biomass. In grassland it's something like 20 times greater. I don't know if you only get stocking densities of 2,000 sheep per hectare, but that's certainly what's below the ground in terms of living biomass. It's huge, and it's processing a lot of stuff, it's storing a lot of carbon, it's cycling nutrients, it's making structure. And so very often you'll see studies where you go no further than this, and this is the overall biomass of the soil, but that's divided into bacteria, fungi, algae, which I'm going to concentrate on, protozoa, nematodes, insects, mammals. And in Central America there is a burrowing budgerigar. Fantastic. It's a soil organism as far as I'm concerned. And of course plant roots. These vary in size from the very, very small to the reasonably big. These are, so in that same sample you'd get anything up to 10 billion bacteria in a teaspoon of soil, 50 kilometres of fungi in your hand like that, tens of thousands of protozoa nematodes, and then a few obviously as you get bigger. You don't actually get 0.01, you know what I do, being a scientist. And that represents a large number of species, and a conservative estimate is 10,000 species in that handful of soil. There are some estimates that there might be as many as 1 million or even 10 million species in a handful. One of the problems with bacteria is that they are horribly promiscuous and will swap 40% of their DNA with a completely different species at will. That's why they do so well. I've got some lovely photomicrographs here from Carl Ritz, former colleague at Cranfield. He's now at Nottingham as we use these. This is a bacterial colony, and as you can see these are filling the pores here quite extensively. They've found a rich source of nutrient one way or another, and they've grown like mad. Of course bacteria grow very fast. Fastest growing organism on the planet is, as far as we know, is Escherichia coli. We've got guts full of them, and they'll double their population size in 20 minutes. Or you can have them where they're quite more sparsely distributed here. And then this is fungal hyphal networks, and they are binding together soil particles. Incredibly important for soil stability and keeping soils open. And this is a this is a nematode. Oh, beautiful. And you can see that that's occupying a pore. And then my favourite, this one, that is a single amoeba moving from one pore space to another through a tiny little hole. and it, of course, will be moving, hoovering up bacteria and moving nutrients from one system to another, and then an earth that will come along, and so on and so forth. These aren't small organisms. This is in the area of a fungus which has germinated from a single spore somewhere in the middle of this. This is Armourilla bulbosa in Michigan. This was the estimate at the time. What's the largest? Come on, it's the QI question. What's the biggest organism on the Earth? Please say the blue whale. Yeah. Okay. Yeah. No, not blue whale. Blue whale's tiny in comparison to a single fungus. And last year I heard that they'd upped that estimate to this single fungus covering 87 square kilometres. That's one fungus. And of course, as Alex said earlier, these then connect up all the trees of the different species in this amazing network. One of the few bits of science that Avatar actually got right. In a complex, in a very, very complex system. So soil biota is very data rich. I like that animation, I'm going to do it again. There we are. So you get lots and lots of data from this. And in fact, it's an incredibly target-rich environment for trying to learn about what's going on. So very quickly, I'm going to show you an example of the kinds of things we've been doing over the years following restorations. This is Abbott Hall Farm in Essex, and essentially this is what's known as a managed realignment site, where part of the site has breached the seawall to try and restore salt marsh. It used to be called managed retreat, but then someone decided that it wasn't Churchillian enough, so it had to be called managed realignment after that. And essentially what's happened is that this sea wall built by, they're fantastic, these sea walls built by a Dutch engineer called Vermuyden. They've lasted for hundreds of years. You actually have to knock them down. They'll be there, well, long beyond our time. And essentially the idea was to say, okay, here's this was just breached after this photograph. We were comparing it with the older breach, the reference site here, the younger breach, something which was salt marsh 300 years ago and something which has never been salt marsh. And essentially we've done this thing where we've looked at the whole community by looking at a range of biomarkers, producing a fingerprint, if you like, of that community, lots of peaks of cell wall markers. And here's the reclaimed farmland 300 years ago. So that was put behind the seawall 300 years ago, and this is the farmland. This is slightly different. This is the 2002 restoration, that's the 95 restoration, and that's the salt marsh. And it fits on a satisfying kind of trajectory. We find this all the time in restoration when we can put chronosequences together. And we can also use this kind of work to discriminate between different land uses. This is arable, red, all on its own, Johnny No Mates, for various reasons, because you've stripped out all of the feedbacks that are going on in these other kinds of environments. So I'm interested in rewilding. I've had a long association with ecosystem restoration. This is a definition that I helped craft many years ago, which is now adopted internationally as the standard definition of restoration. Importantly, it's a process of ecosystem restoration. It's not something you do and walk away from. And importantly, the strapline to this is the reconnecting nature and culture. So people get very obsessed with the technical aspects of this, but part of this is how can we reweave our interactions, connections with nature. This is a diagram that I produced years ago with Richard Hobbs, where essentially we're looking at intact ecosystem states and fully functional ecosystem attributes. And if you've got a very degraded site down here, to get up here it's difficult, but you have to cross two barriers, an abiotic barrier and a biotic barrier. And there'll be aspects of this at Knepp, but also wherever you go, you have to get the abiotic conditions right before you can really start playing with the biotic conditions. So this is where we sampled adjacent farm. We got the ancient woodland, surrounding Wagstaff's wood, rainbow field, so on and so forth. The reason we got these is we wanted a range of times since rewilding had started. Lots of soil biology, microbial biomass function, a phenotype and some soil chemistry. And so I won't keep you in suspense any longer. This is what we found for soil carbon. Wow, look at that. There's arable at 0 to 10, 10 to 20, 20 to 30, and we've got at least a doubling of soil organic matter over that time. I'm going to show you two other versions of the same thing just to show it doesn't matter how you measure it, you're getting the same result. But importantly, if you look at the 2005 one here, as you go down in depth, you're getting soil organic matter going all the way through. With the arable, it's kind of doesn't increase, and in fact starts to tail off. In the rewilded site, it started to increase. This is, we've had to put a little break there because the woodland or forest site has such high organic matter, but I'm going to talk about that as a reference and a target in a moment. Same with organic carbon through a different measure. Again, this enormous difference between the arable and the rewilded sites. And even more spectacularly, a trebling of the microbial biomass in many of the rewilded sites, and they're all higher than the arable and at depth and approaching this forest. Now, I don't think you can simply use that woodland forest site as a reference, because we don't know what the final stable state of a rewilded system is. So it's something we can compare it with, but we're not saying that that's the target. I think that's really important that we understand that, because otherwise you can get led up all sorts of garden paths about, oh, we're trying to achieve it, so it looks like that. I don't think we know enough about rewilded sites and rewilding and semi-natural sites and natural sites to say it should look like that. I really don't. I think what's important here is we've reinstated some natural processes that have been lost. Very interesting, fungal biomarkers. We've got two here. They're more or less the same across here from the arable, but look at this one. This one shoots up in comparison to the arable site, and again that's linked to certain sorts of fungi. We don't know exactly which, but importantly it plays out in this fungal to bacterial ratio, very low fungal component of the total PLFA marker signal, which in the rewilded sites, especially when you get back to 1999, has gone up significantly, even more than the woodland. So this means that the system's shifted from being a bacterially dominated system to one that's being dominated by fungi. This is very complicated now. This is the phenotype, and interestingly, the phenotype of the arable and the woodland are very similar, but they're very different from the rewilded site. So there's been a significant shift in the community, and this is why I'm cautious about using this as a reference site. We simply don't know what the eventual quasi-equilibrium system will look like. But when we look at the ability of the soil to process a wide variety of carbon substrates, we find very clearly that they're starting to move sort of in the direction of the woodland. There's no difference in this component, for example, between that and this, but there are differences. So anyway, we're moving away and moving more in this direction. What's important is the catabolic capabilities of the soil microbial community is changing. So the woodland isn't necessarily the target. And so what I wanted to do, finish and put this in the context of the wider dialogue around what we want with our land use futures, what are we trying to do in terms of having a successful relationship with the land and society. This is work with a very good colleague of mine, Eric Higgs, who I publish with all the time, is looking about the way in which you might classify management intention, ecosystem-centred, human-centred, and the intervention, whether it's a composition or a functional intervention. And you could say that you've got kind of green infrastructure for urban areas, agroecological reclaimed systems, and then novel ecosystems that we've been working on. The question is, where would you put rewilding on that? I've got a view on this, and the idea is you've got self-assembled and designed systems, and the idea is if you restore a system, there's been a huge discussion about historical systems and what we're aiming for. It's just impossible to do. We cannot go backwards. We can only restore forwards. We've got a change in climate. We're going to have to make systems ready for the future. And one of the ways that we can do that is make sure that there are natural processes which allow the system to respond ecologically and evolutionarily. So I think that rewilding might be fitting here. There is some design in the system, there's no way around that. There are management interventions in the system, but there's a large amount of self-assembly. It may be that they are fitting in these emerging ecological approaches where there's more design in it, and then they become novel or hybrid. They're not become historical, nothing is. Or perhaps they'll some kind of hybrid system. Why is this important? Well, the reason for that is that we need to come some kind of view of the way in which we're managing landscapes, the way in which it engages and benefits with society, the way that there is some information from the past. And you've got a prime example here with the work of Vera, who's influenced what you've been doing here by talking about those Pleistocene type landscapes, the ecological integrity of the system. We've seen loads of examples today of that kind of thing, that reweaving of nature coming in to the site here. And long-term sustainability, and it's being sustained. It's not constantly requiring high energy material inputs to the system. So the emerging discussion is around the circular economy and the way in which we can try to reduce our inputs. We've got industrial agriculture, monoculture, lots and lots of turning plough, fertilisers, conservation agriculture, crop rotation, no-till, heavy use of herbicides and use of chemical fertilisers. So that's a step to try and reduce that inputs. And then we've got this whole swathe of practices which are aimed at producing outputs such as increase in soil organic matter, everything from permaculture through organic, no-till, holistic grazing, and actually producing terraces and what have you. So the thing about this is that this is happening right now. And as I was saying earlier, certainly in the UK, we're in an era where public policy is the most plastic it's been for the last 60 or 70 years, and there will be a window of time, not going too far in the future, where that plasticity will stop and we'll be back with another 50 or 60 years before the intensity and the breadth of the discourse is as it is now. What do we want to do for the future? So, we think we can improve soil health. Certainly the data that we've got kind of suggests that what's happening at Knepp with rewilding is starting to do that, although I've got a problem with the term health, but I can talk about that later. And importantly, we've got this crossover from production to semi-natural, which I think is where rewilding is, but certainly it's on that right-hand side of the, is it the virtuous part of the spectrum? I don't know. And it's consistent with the 25-year plan and what's in the environment bill, as indeed Knepp is mentioned in the 25-year plan. Internationally, things such as the IPBES, which is the Biodiversity and Ecosystem Service, sister of the Intergovernmental Panel for Climate Change, is suggesting different scenarios of what might happen. This is the land degradation neutrality scenario, with rehabilitation and mitigation being very important. This is the idea we protect more and rehabilitate it, and that's that fits. That's where the net might fit in both rehabilitation and mitigation for the future. So, doubling of soil organic matter, similar trend for microbial biomass, even greater increase in general fungal biomarkers, and rewilding can become an essential part of how we manage the landscape. Thank you.